All right. Well, hi, everyone. My name is Chrislyn Particka, and I'm the host and moderator of the National Clean Plant Network Grapes webinar series. Just a little note, this is not a live recording. When we were doing the live webinar, we had a lot of audio problems. So Josh has agreed to redo his slides, and then we were able to capture the questions that were asked during the live webinar. So Josh will answer those questions again at the end. So anyway, Josh, I'll let you go ahead and get started.
Thank you. As indicated, my name is Josh Puckett, and I am the production manager at Foundation Plant Services UC Davis. Tim did a great job of discussing the impacts of virus on commercial vineyards and the industry as a whole, and the importance of clean plant material. And what I'm going to talk on through the next set of slides is the perspective from Foundation Plant Services and the components required in delivering clean grape plant material to the industry. If for more information on anything that I discuss in these slides, or for information on topics not discussed, you can visit our website at fps.ucdavis.edu. Foundation Plant Services is a self-supporting, service-based department in the College of Agricultural and Environmental Sciences at UC Davis. Our mission is to produce, test, maintain, and distribute elite disease-tested plant propagation material, and we do this for a variety of crops aside from grapes. But the important thing is that each of these crops is produced and maintained by vegetative asexual cuttings. So the importance for starting with clean plant material is very high. Foundation Plant Services also provides custom importation and quarantine services and virus testing and elimination. We are the major import facility in the United States for grapevine. And we coordinate the release of UC patented horticultural varieties for grapevines, fruit and nut trees, and strawberries. And just being in the position that we are, the mission of Foundation Plant Services is also to serve as a bridge between researchers, nurseries, and producers in each crop industry. Material is introduced both domestically or from foreign locations to FPS as dormant grape cuttings. From that material, we are able to then establish mother plants, which are used for treatment and testing. We perform a number of different testing techniques, which I'll get into in a bit, but the general goal for this testing material is to identify clean plant material, or to produce it through treatment, establish it in our foundation vineyards as provisionally registered, and confirm it to be registered through ID verification by ampelography and by DNA fingerprinting, and then be able to distribute to nurseries and growers virus-tested treated material. There are a number of government regulations in place by federal USDA and by California CDFA that maintain and regulate this, what they call the grapevine RNC. That means Registration and Certification Program. There are sets of viruses that are required to be tested for at the foundation level, which is what FPS is. And then there are sets of requirements that are less requiring for the nurseries that receive that material and maintain primary or secondary increase registered blocks. And those are the blocks that they are able to then grow out the Foundation Plant Services material in large enough volumes to supply acreage-sized plantings of certified nursery stock. And on this slide, there's just text I've pulled in directly from the CDFA requirements that indicate that in addition to the initial virus screening, the foundation level material is required to be inspected twice each year. Us, the USDA and CDFA, walk through the vineyard blocks and look at every vine. And then we are also required to retest every plant in the foundation within a 5-year period. So what we do is 20% of the collection is tested, what we call maintenance testing each year. Then the requirements you'll see on this slide for primary and secondary to the right increased blocks are less requiring. They only have to test for a number of vineyards. And at this point, it's not every single vine. It's a representative sampling, and that's within every 5 years. For more information, I put a link at the bottom of the slide that could take you directly to the CDFA website. So the strategy at FPS for pathogen detection is to employ a number of different broad and specific-based test methods. We use herbaceous host indexing, woody field indexing, ELISA, real-time PCR, and recently we've incorporated high-throughput sequencing. This is a new technology that will be coming more into play in the coming years and possibly allow for reducing reliance upon these time-requiring herbaceous and woody indexes. I'll go into each of these tests now in a little bit more detail, but not too much. The woody index is basically, we use 3 indicators that have been demonstrated through various historic research to show very clear symptoms for specific viral agents. And we start with clean plant material in our collection. So the goal is to chip bud the candidate material, we call it, the material in question, whether domestic or foreign source of introduced material, onto these indicators, plant them in the field, and then after 2 years, we inspect them for leaf and trunk symptoms. So this slide and the next will just show you a little bit more of what that process entails. So we pull material from our foundation vineyard in 18-inch cuttings, disc bud it, wax it, stick it in a vermiculite perlite media, initiate root growth and shoot growth, and then we chip bud the material that we receive onto that. We do, for each indicator, 6 plants just to give us a larger sampling size. The important part of the test is to ensure that there's connection between the candidate material and the indicator at the cambial layer. We make a notch and pull out the bud of the candidate and make a similar size notch in the woody indicator, and then we wrap the bud to ensure that it doesn't desiccate. We will then read the bud take of these after 2 to 3 weeks in the greenhouse. We will move them out to the shade house, allow them to harden off, and then plant these in the field. And again, we'll read the index at this point 18 months later. The 3 indicators that we use are Cabernet Franc, LN33, and St. George. In Cabernet Franc, we're looking for red leaf symptoms in the fall, typically for the leaf roll viruses and red blotch. In LN33, we're looking for the presence of GVB. It causes corky bark in the canes. And the picture to the right, you can see an example of a healthy cane on the top and a cane showing corky bark symptoms on the bottom. And in St. George, we read in the spring, sorry, the LN33 is red in the summer. The St. George is red in the spring for nipoviruses and fleck. And you can see what a leaf showing fleck would look like to the bottom left of the slide. And typically, it's not required by the CFA anymore, but this St. George, being Vitis rupestris, has been a indicator for rupestris stem pitting, and that's another rugose wood symptom. Our herbaceous indicators and index rely upon 4 different herbaceous hosts. We use Chenopodium, Amaranth, and Quinoa. We use Nicotiana clevelandii, and we use the cultivar National Pickling of cucumber. For this test, unlike the woody index where we're testing graft inoculation, we're testing mechanical inoculation here. So we agitate the leaves with carbonium powder, and then we take sap, basically ground leaf tissue, with a nicotine buffer and apply that to the surfaces of the leaves. We rinse that off with water, and we'll read that within 7 to 21 days. And on this slide, you can see examples of what a healthy versus virus-infected leaves would look like for cucumber and for the Kochiana. The other more specific laboratory diagnostic techniques that we use are ELISA, which is enzyme-linked immunosorbent assay, and then we're testing for presence of antigen, viral antigen, or proteins, and PCR, polymerase chain reaction, where we're looking now for specific, we can get to the strain level, for example, of the leaf rolls, which the slide shows. We're looking for specific conserved nucleotide sequences. So the goal of employing each of these different strategies is that we have a broad perspective of disease using the biological assays, the indicators. Those don't tell us what specific pathogens are present, but they do show you a visual cue that something is present. And then we're able to look then a little bit more specific using ELISA and even more specific using PCR. Any one positive of using any of these techniques would be enough to not allow that selection to advance forward into the fields. So when we do identify virus-positive selections, the technique that we use for eliminating virus is microshoot tip tissue culture or other meristemmed shoot tip culture. And what this entails is taking a 0.4 millimeter section of the apical meristem, which you can see at the top left, showing you the apical meristem, and then to the right of that, what we would collect from the mother plants that we've established with incoming material. Under a dissecting scope, we would then remove the outer leaf layers, identify the apical dome, which in two-leaf primordia typically, excise that and establish it in vitro in media. And this media is basically a combination of sugars, salts, and hormones. Every 2 weeks, a technician needs to take the plantlets that are in a tube and transfer them to a new tube because those plants will exhaust the nutrients in that media. And the bottom of the slide, you'll see that indicates that this is about a 6 to 12 month process. And to give you a perspective of the volume that we're working with here at Foundation Plant Services, I've shown what the last 5 years have been. In 2011, 156 selections were excised, 2012, 124, and we've continued at that pace for the last 5 years. I've chosen these 5 years because as Tim mentioned, National Clean Plant Network money, which became available as a result of the 2008 Farm Bill, has really allowed us to expand the volume of selections that we're treating and move into our new 2010 protocol, which I'll touch on at the later part of this discussion. So once we've established a rooted multi-node X-plant in vitro, we'll need to transfer that to soil. So we'll take that out. We have to remove the media and establish that in an autoclaved, sterilized soil. We take that to our environmental growth chamber and try to then transition it from the optimal dependent conditions it was in vitro to the realistic conditions of the shade houses and ultimately the field, if we're able to confirm through testing that whatever was present in the mother plant material has been eliminated. So it is important to note that I mentioned that the biological index can take up to 2 years, that the tissue culture treatment can take up to a year. One thing we have adopted into our protocols to try and reduce the time that this process takes is that as soon as material comes in, before we have the final results, we'll initiate tissue culture. But that being said, if something goes through the biological index, comes up positive, then we have to subject that treated plant to the same tests, biological and laboratory, to confirm that we get negative results to replace all the positive results of that mother selection. So this is a very busy graphic, but if you look at the top of the slide, it shows you that the successional movement of this material and how this can very easily become a multi-year investment. If something comes in clean, we're able to, within 3 years, get that out into our foundation and have small amounts of material available. If we have to rely upon a tissue culture plant, this could become up to a 6-year, maybe more project. And once these are established in the foundation vineyard, of course, at first it's very limited material, and it remains limited what we are able to provide. But what we do distribute are dormant cuttings. We sell them based upon the diameter. So you could find all this information on our website. And then we'll also, in the initial years when we don't have available budwood for grafting, we can make 2-node unrooted propagations, which we call MPPs, and these are available for purchase. This has really been the model we've gone with for the new high demand selections, getting them out into the hands of the registered nurseries as MPPs, allowing them to be the source of their own budwood, and then they can quickly graft their primary and eventually secondary blocks to increase or decrease the amount of time it takes for them to provide certified material to growers. Here's a kind of a graphic demonstrating what that would entail. We are able to provide these as MPPs and typically in small quantities. So that using this example, we're just taking what one plant would through the course of multiple years amplify to from a nursery perspective. And so I mentioned our process could take 3 up to 6, maybe more years, and then there is a period of time required between then and the point to which the nursery has that selection, that source available for wide-scale distribution. So there's been a lot of excitement about 2010 protocol and Russell Ranch. Little history on what that is that from the 2008 Farm Bill, FPS was asked the question, if you were to create the next level of standards for clean plant material, what would that require? And so in 2010, a protocol was developed, and plantings were first put in 2011, and we've continued since through 2015. And what that protocol entails is we know that micro-shoot tip tissue culture, the therapy I mentioned earlier, eliminates known virus. So the thought being that as we bring on tests like high throughput sequencing, or as we improve diagnostic techniques and are able to more easily identify new viruses, perhaps we have, through micro-shoot tip tissue culture, we're able to produce plants that are free of currently unknown virus. And then we've also added to the tests that we already perform, in addition to the herbaceous and woody indexing and ELISA, we've added multiple PCR tests, and currently for the 2010 protocol, we're up to 35 tests that are required. And that number continues to grow as new viruses are identified. And so this slide here just gives you an idea of everything that any vine in Russell Ranch, which is every vine in Russell Ranch is of 2010 protocol, they've all been tested by these standards. And included in this, of course, is red blotch, is the obvious example where we confirmed that when 2010 protocol was established, we didn't have information on red blotch. But since the development of the primers, we retested everything in Russell Ranch and identified no presence of red blotch, and we associate that with the preemptive micro-shoot tip tissue culture. So as I mentioned, first planting, 2011, we were able to put 208 selections in 2011, and we've kept at that pace. We've increased in selections each year. In 2015, there were 355 selections. The land at Russell Ranch, I'll mention, is 100 acres that we were granted from the university, and currently we're occupying about half of that with grapes. So another goal of the 2010 protocol was to elevate the standards without losing the industry recognition of particular selections in our collection and of federal the collections that are out there. And so an example here I provide is that Chardonnay 04 is a well-known and popular Chardonnay selection, and the 2010 protocol, so it had not been through microchip tissue culture. We submitted it to treatment, tested it by 2010 protocol standards, confirmed it to be qualified, and established it at Russell Ranch as Chardonnay 04.1. So the 0.1 indicates that it's gone through 2010 protocol treatment and testing and is at Russell Ranch, but we now know that we can maintain that it was sourced from Chardonnay 04, so people can recognize that. This selection and most of the selections in Russell Ranch Scion selections are represented only as 2 vines, so the material is quite limited. I've tried to provide a guesstimate on what would be available from those 2 vines each year. So 2011 planting is our oldest planting, and I would anticipate that we could, from the 2 vines, pull maybe 30 number 1. So number 1 is a cutting that is larger than 1/4 inch in diameter, 30 number 2s, and 20, that should say, number 3s. Our rootstocks are represented based upon popularity from 2 up to the most popular having 12 vines out in the field. And our major effort was to initially distribute those rootstocks, because it's important to note that if you want to maintain a vineyard at that 2010 protocol standard, you want both a 2010 protocol rootstock and scion. So the thought process there was to first get the rootstocks out for people to graft upon. And again, the availability largely depends upon the vigor of the crop. I've tried to show you what you could anticipate based upon the development of the vine, and then the demand for this material is also what really limits availability. It is extremely high, and we are working with a limited amount of material. To find out what is available, selections, we have on our website we have 2 lists actually. You'll see that we have a list called available selections, which indicates selections that have been in the ground for more than 3 years, and then new introductions. And we split the 2 just to so that people could have realistic expectations of the availability of material. This again can be found at fps.ucdavis.edu. Across the top runs a blue bar with links for each crop and service that we provide, and as you click on those, it changes the options in the menu to your right, the gold menu. And with that, I thank you for listening, and I will take the questions.
Okay, Josh. The first question was from Lena, and she asked, do you see vines infected with more than 1 virus?
Yes, yeah, I remember the question. Yes, we typically, when we are screening new introductions, will find that there are multiple infections. So it's actually more rare that we find vines with a single infection.
Okay, and then she had a follow-up question that we addressed later, but I'll go ahead and talk about it now. She asked, what do you do when you find a plant that does have more than 1 virus?
Yeah. So what makes microchip tissue culture an ideal tool for eliminating virus is that it is not virus specific in what it eliminates. The viruses, typically these grapevine viruses are phloem limited, so they're moving systemically through the vascular tissue. And the goal with the microchip tissue culture is to take a small enough excision to where we don't risk any kind of mutation to the cultivar. Large enough to where we don't risk mutation, but small enough to where we're not bringing in any of that vascular tissue. So we should, in theory, be eliminating all phloem-based viruses.
Okay, and then Cindy asked if FPS is the only NCPN center with an import permit for foreign grape varieties.
No, and we had some discussion about this in the webinar. Cornell does have permitting for that. It was mentioned that potentially Missouri did, and I would check with Washington State University, the Prosser Center, Northwest Clean Plant Center.
Okay. And then Cindy also asked if FPS is beginning to do next-gen sequencing on new varieties.
So currently, I want to say about 2 weeks ago, we were able to purchase a department deep sequencing machine, an Illumina sequencer, and we've I think probably gone through about 5 different samples for grapes. We're currently in the protocol development stage, though, where we're trying to optimize the sampling, the processing, and the technique to where this will likely, and we're thinking this is a 2017 type of endeavor where we'll start performing the high throughput sequencing on incoming introductions in the next couple of years.
Okay. David asked if the new Pinot Gris virus is on the FPS list. Yes, the new Pinot Gris virus, we have added it to our protocol for our 2010 protocol testing, but currently we have only been able to, since it was just recently identified and even more recently identified within the U.S., we have only made it through about 1/3 of our Russell Ranch vines in testing. We're going to, in the next year, try to bring every vine current with testing, and the maintenance testing that I mentioned, in which we test 20% of the vines, we're going to add it to that panel. So with the Classic Foundation, we're not going to immediately have results for everything as we're trying to for the Russell Ranch, but we will work towards within the next, and we did add it to last year. So we have, I believe we're actually ahead where we got about 40% of the Classic tested, and then 20% will be tested each year going forward. Okay, and then David also asked, in addition to viral monitoring in the Russell Ranch Foundation Vineyard, what kind of vector and insect monitoring is done? Mm-hmm. So being a foundation level planting ground, we still have access to methyl bromide, which has allowed us to fumigate all soil prior to planting, which reduces the threat of nematode vectoring. We are also lucky to be very isolated and not surrounded by commercial grapevines. But also, we do work with the county agricultural office to do insect monitoring, and we are very frequent with our pesticide applications to prevent any kind of incoming or secondary movement if it was to occur. Okay, and then Andrew asked if you have seen any mutations in the tissue cultured material. No, we have not, and I think that the idea of mutation through tissue culture, although still maintained by some, has been put to rest. Since the webinar, I got more information, and that there were some papers that were produced in France, and none of them were successful in demonstrating mutation as a result of microchip tissue culture. And what I mentioned the previous week is that we've done some work with Ian J. Gallo, where we provided them untreated and then multiple treatment samples, and those were compared in the field for by production, and the fruit was analyzed to see if there were any differences, and none were identified. Okay, then Daniel asked if you could talk about the viruses that have been discovered since 2010. Yeah, the major virus that comes to mind is red blotch, and again, we were very pleased to find that when we did a full sampling of every vine in Russell Ranch, that we identified 0 positives. We did perform the same sampling in our foundation, again, the difference being that Russell Ranch has all gone through microchip tissue culture, and the foundations, some have, but not all, and we identified 15 cases of red blotch positives. Those have since been eliminated, but it demonstrated to us the importance of having the preemptive microchip tissue culture as a part of our protocol. The Pinot Gris virus was also mentioned, and there have been a number of recent viruses. The issue here being that we're in an age where
detection and identification of genetic sequence is becoming quite easy, but the understanding of the implications of that virus or the biology of that, that takes much more time to develop. And so we do what we can. We're preemptive, where we're hoping that the initial treatment and then eventual adoption of high throughput sequencing will allow us to have vines as clean as possible. But one thing that may come from all this high throughput sequencing is that we identify that some viruses are just ubiquitous, not very economically impactful, and we'll realize what the practicality of clean material is.
Okay, and then Lena asked if there's any difference in susceptibility between vinifera and hybrid cultivars.
To virus?
Yes.
Yes, and we know that there is through rootstock, and there have also been in the hybrids demonstrated to be differences in, and I would use it more as, yes, susceptibility or tolerance, not saying resistance, because these can serve as a reservoir for the virus. They're less symptomatic. The phenotype seems to be less impacted by the virus, but it's not to say that the virus is incapable of establishment and then moving systemically through that vine.
Yeah, okay. Then there was another question, is there any evidence of propagation through pruning? So when you're out in the field pruning and a virus from one plant gets on the pruning shears and then it gets passed to another plant?
Yeah. With grapevine viruses, we have not found a case where mechanical transmission. Through grafting, certainly, but these are typically phloem-based, and so the vectors of these tend to be phloem-feeding either insects or nematodes. And with our other crops, prunus, certainly this is an issue, and for viroids and phytoplasmas, this can be an issue as well, but for the grapevine viruses, no. Okay.
And then the last question is, when is the best time to scout fields for the presence of viruses?
Yeah, so our biannual visual inspections are scheduled in the spring and in the fall, because that would be the best time, just as I mentioned with the indicators that will read those newly emerging leaves. Those will show symptoms well, and then in the fall you'll get a good view. Once the temperature elevates and it gets warm, the plant will be able to grow out, in some cases, the visual symptoms, and so it's more difficult to identify in rogue if that's your strategy.
Okay, well thanks Josh. We made it through all the questions. I'm glad that we were able to redo the recording because the audio quality is perfect this time. So thanks for that. Let's see here. Tim did some acknowledgments at the end of Joshua's talk, so I'm going to go ahead and go over those now. First are the speakers and the people who are involved at Cornell University, and then also Josh at UC Davis, plus Sue Sim and Deborah Galino, who both work there and are also involved with the National Clean Plant Network. Upcoming speakers from New York Department of Ag and Markets, presenters at other universities. Our last webinar is going to involve 3 nursery owners in New York. And then finally, some leadership within the National Clean Plant Network. Eric Reedy-J, who's at the USDA APHIS and coordinates the NCPN program. And then Rose Gergich and Keith Striegler, who are the current outreach coordinator and former outreach coordinator. Then there was funding provided by the USDA APHIS, National Clean Plant Network, as well as some money from the New York Farm Viability Institute and a grant through the New York State Department of Ag and Markets Specialty Crops Block Grant Program. So with that, I think we're all done, and thanks again, Josh.
Yep, thank you.